Welcome to Concept Biology. I'm Alex. In today's class, let's explore the fascinating process of spermatogenesis, how sperm cells are formed inside the male body. Spermatogenesis takes place inside the seminiferous tubules of the testes. Here, special cells called Sertoli cells and Leydig cells provide sport and hormonal control. Within each seminiferous tubule, sperm cells develop step by step, starting near the outer wall and moving towards the lumen as they mature. In the first phase, called the multiplication, diploid spermatogonia divide repeatedly by mitosis. Some remain as stem cells while others prepare to form sperm. One of these cells enlarges and becomes a primary spermatocyte. This cell now enters the growth phase, ready for the next stage, that is meiosis. During the first meiotic division, a primary spermatocyte divides into two secondary spermatocytes. This is called reduction division, where chromosome number becomes half. Next, each secondary spermatocyte divides again, forming four haploid spermatids. These are immature sperm cells that still need to differentiate. In spermiogenesis, each spermatid transfers into a mature spermatozoon. It develops a head, a middle piece packed with mitochondria, and a long tail for movement. Finally, the sperm is released from the Sertoli cells into the lumen of the tubule. This release process is known as spermiation. Each sperm has three parts: the head, containing nucleus and acrosome, the middle piece, rich in mitochondria, and the tail that helps swim towards the target. The entire process is controlled by hormones, that is GnRH or gonadotropin releasing hormone from the hypothalamus stimulates FSH, that is follicle stimulating hormone, and LH, that is luteinizing hormone released from the pituitary, acts on Leydig cells to produce testosterone, which drives spermatogenesis. To summarize, from one spermatogonium, four haploid sperm cells are formed through the series of mitotic and meiotic divisions followed by differentiation.